Hi, Dr. Brian Kaufman, a retired family doctor, a CLL patient myself, and the Executive Vice President, Chief Medical Officer, and the co-founder of the nonprofit CLL Society. Excited to be back live at ASH 2022 in New Orleans.
Hi, my name is Neil Kay. I'm a hematologist working at Mayo Clinic. My focus is CLL, and I'm also glad to be back at ASH and to be here with Brian.
So, you and your colleagues at Mayo Clinic are presenting some research on, and I'm going to read this because you're going to unpack it for the patients here.
Okay.
Differential susceptibility to senescence in CAR T-based cells based on co-stimulatory signaling. So, explain to someone who has no idea what any of those words mean, what you're researching and why should it be important to a patient?
Thank you, Brian. So I want to make it clear, by the way, that this is not research that I did primarily myself, but I'm in deep collaboration with Dr. Saad Kenderian and his group that are going to be presenting this abstract. This is building on an immunotherapy approach where, if you will, genetically manipulated T-cells are able to, via the genetic manipulation, home in and literally be like a drone attracted towards a CLL B cell, and to then, because of the nature of the T cell, where we're hoping it's viable, functional, and cytotoxic, a killer cell.
So cytotoxic means it kills cells, yeah?
Yes, yeah. Can effectively reduce the leukemic burden in a given CLL patient. Now, I'd also match that with the unfortunate knowledge that the immunotherapy, or the CAR T that we just talked about, this genetically manipulated.
Can you remind us what CAR T stands for?
Yeah, it's a chimeric receptor that's genetically placed in a T cell, where it's part of the T cell receptor that's normally on T cells. Plus, it also has a component that is that's able to recognize a receptor on a CLL B cell. So this receptor allows the homing and also potentially facilitates activating the cell to become a killer cytotoxic T cell.
And chimeric just means that it comes from something outside of a human, that it's been genetically modified for us to tolerate it and recognize markers on the CLL cell and hone in, like you say, and attack them. Yeah.
And there's some very well-established genetic manipulations that can be done to introduce a variety of different types of receptors into these T cells.
And just to set this up, CAR T, I'm 4.5 years out plus CAR T therapy, but that's rather exceptional in CLL, to be honest. Most patients haven't done as well.
Exactly.
And unlike some of the acute leukemias and even some of the chronic leukemias, we still don't have an approved CAR T therapy.
Excuse me. Yeah.
And we've had less success with CAR T, arguably, than a lot of other cancers. Is that what this research is about, trying to figure that out?
Yes, exactly. And unfortunately for CLL patients, this approach would not be uniformly effective. It does work in given patients. Hopefully for you, it did was a significant health benefit. Excuse me. However, most CLL patients, there is not that vigorous response. And Dr. Kenderian's lab and his team are trying to unravel why that is. And there's a variety of things that could be going on. But this investigation was specifically looking at the possibility that these T cells might be becoming senescent.
So explain what senescence means in a cell.
So senescence effectively is saying that this cell is not functioning normally. It may be, in fact, a little bit much more of a tendency to be inflammatory in nature, and also that it is not capable of killing to the same level as a typical mature cytolytic T cell.
And are there findings that you can share with us that from the lab in terms of what this means in a patient-friendly way?
Yes. So the manipulation that was done here was to study different types of these genetically modified receptors, and there have been several generations of these that have been developed over time, and to determine whether those types of genetic receptors over a series of activations that occur in the lab, this is all in the laboratory, all preclinical, would end up generating this type of senescence. And so this was a very elegant piece of work where these T cells were genetically modified in 2 or 3 different ways with these different receptors, and then activated at different time points, I believe at day 15 and day 22 in the laboratory. And then ultimately they were looked at for senescence markers and also for the ability of these T cells to reduce tumor burden in mice models. Excuse me, we have to cut this out. So the outcome was that for a specific kind of receptor, more of the first generation receptor, and also with multiple activation time points, particularly later time with day 15, day 22, there tended to be evolution of what looks like a senescence T cell, and when these T cells were put into mice that were given tumor to deal with, the first generation T cell, the one that had been activated at a later time, was less effective in the mouse model. So it looks like one of the maneuvers that may have to be overcome in generating more effective CAR Ts is to avoid the appearance of senescence.
So are there ways your colleagues are looking at that we could maybe tweak we're genetically modifying these already. Is this looking at just another fix genetically to make these more active, make them less?
There are approaches to make them more effective. And in this particular abstract, this was more a definition of, is senescence there or not? It appears to be there. There are ongoing activities now that have to do probably which genetically modified receptor do we use, and what is the level of activation that might be, what's the optimum time to activate these T cells. I should mention that, by the way, they're activated by exposure to malignant cells. So that there are some approaches that could be developed based on this, but is not reported in this particular abstract.
Any final thoughts for patients on this particular subject or on CAR T more generally for CLL?
Yes, I have high hopes for CAR T in immunotherapy for CLL patients. We have known for a long time that activated T cells can effectively kill CLL B cells. My intellectual bias is that we will ultimately be using these relatively frequently, but probably for patients who have been effectively treated initially with novel agents, for example, and then, if you will, remove the remaining T cells using this approach, because these CAR T cells are able to go anywhere and to effectively remove disease in lymph nodes, bone marrow. So I'm hopeful still, even though it's been a while now that they've been worked on, where it will be applied to even potentially move us closer to a cure for CLL.
Well, I'm certainly a believer in them and excited about them, and can't wait to see them tweaked and their efficacy improved, because I am kind of an outlier, and I'd like to see more people to be able to follow in that footsteps. We just haven't had the success in CLL.
Amen to that.
Yeah. Dr. K, thanks so much.
My
Great
pleasure.
to see you again.
Nice to see you.